Dr. Elizabeth Blackburn joined Joe Gall's lab in 1975 and started to explore Tetrahymena, a tiny organism with many thousands of chromosomes which live in pond scum.
So I came to Joe Gall's lab in February of 1975, and so we quickly decided what I was going to work on. And I explained to Joe that I was interested in working on the DNA sequence at the ends of chromosomes. And we had a brief discussion about the kind of organism that might be the best one to work with. And there was another kind of organism which was related to the ciliated protozoan Tetrahymena, in which Joe Gall had found large numbers of very short chromosomes, linear chromosomes. And this other related organism had such chromosomes, but Joe showed, this sort of scientific acumen. He just said, go for the one that you can grow easily and just, don't have to spend a lot of time trying to get the organism to grow. So I chose Tetrahymena on his advice.
Protozoa are these wonderful single-celled organisms that swim around in ponds and lakes and streams everywhere all over the world. One of these protozoa is a very nice organism for studying in the laboratory. It grows very rapidly. It grows like bacteria. You can grow up a huge colony overnight. You can get lots of material for biochemical analysis. And we were interested in the DNA molecules inside the nucleus of these protozoa.
People knew by the mid-1970s that most DNA in a chromosome is bound up with proteins, and the proteins are called histones, and there are other proteins as well, and those control the way the genetic material is packaged and is also allowed to be expressed so the genetic information can get used by the cell. So of course I wondered, well, was the very end portion of the mini chromosomes also packaged in the same way or not? And so while I was a postdoctoral fellow in Joe Gall's lab, I did some experiments in which I addressed that question, and the answer was no. They were packaged in some fashion that was quite distinct from the standard way that most of the genetic material in a chromosome is packaged. But at that time it was very much a kind of, how do cells work the way they work kind of question. It didn't really have anything to do with, how does this impact on human disease or anything like that. I really wanted to know, why do cells work the way they do? Why does biology work the way it works? It's just an endlessly fascinating question, and this was one way of looking at it.